it's elongated, but there can also be irregularities across the surface. So really what this does to the information coming in is it scatters the focal point. So rather than a single focal point, you kind of get multiple focal points. And so crap looks blurry, man. And so you fix that by a lens that's going to fit the particular astigmatism. Unfortunately, astigmatism does fall into some general categories that can be treated with lenses and contact lenses as well. You know, so we're kind of weighted to try to keep the part where you have your most correction where you need it and so forth. So astigmatism is just abnormal shape for the cornea, not so much for the eye. And so that's one of the differences. And then the old presbyopia. Pres referring to aging. Let's see what is the actual. I love the roots of words. And I know you do too. Oh, I didn't write that down, but pres means age or elder. Okay. Anyway, and it's the change that occurs with the aging eye. And it's really, you know, the eyeball isn't changing shape. It's the fact that you're losing accommodation. But it functions like hyperopia, where you put that, that sounds like a drug, doesn't it? I used to use hyperopia. I smoked it. But the image is behind the eye, and so you move it toward the eye with that convex kind of lens. Presbyopia functions like hyperopia, and you all get there, unless you get there early, and then you're there now. Where am I? Okay, so that's some of the visual deficits. Let's, oh, here's the little lenses: myopia, concave, hyperopia, convex, astigmatism. That's the shape of that one. Just to fit the elongated cornea. There you go. Oh, the best part, the physiology of photoreception. Light strikes the atmosphere and enters your orb, and there is a conformational change of a chemical, and then you can see. I mean, is that just some shiz right there? Some crazy shiz right there? That you go from a wavelength of light to an electrical stimulus that's interpreted as this picture. And no matter how, you learn all this little bit and all these bits of it, and it's just so miraculous to sit back and think about that. And you just, how does that happen? And all we're really seeing is the light bouncing off the stuff, landing in there, and retinal going like this. And just that, and you can see. Love it! Light strikes the pigments of the photoreceptor cell, inducing conformational change. That activates a bipolar cell, then a ganglion cell, and the axon of the ganglion cell will take that information along the optic nerve, cranial nerve number, to the visual cortex. That's it. That's how it works. That's
why you were so
that was someone that just got their grade back from Okay. Oh. This I love. This is the kind of stuff I love, and you'll have to indulge me for a minute because really zoology is my kind of first love. I love biology, but zoology and sort of Earth and just how all this crap fits in. It's just so miraculous because you get lost, you forget about it, and next thing you know, you're just sliding through Instagram. You know, and just not, and you know, when you learn how difficult all this is, you think, I don't even want to look at something I don't want to see ever again. Why would I waste my time? And within seconds of that decision, you're on Pinterest. But that rate that you can see that stuff, and you go, oh, cute shoes. You know, that rate that you're able to do this, unbelievable. Okay, so it's because there's light on the planet, and one of the first things, I mean, gosh, flatworms have photoreception, light-dark reception. A lot of cells do. And so the function of light reception is really the same across biology. And so you remember remember, recall with me now, won't you, photosynthesis, where there was the, what's the big pigment we all really love? Chlorophyll. Oh dear God. Where's my hyperopia? Chlorophyll, one of the many pigments, it appears green, and its function is to serve as a light source, as a light, like a panel, solar panel. And so the chlorophyll is placed in these discs in the chloroplast, the organelle that received the light. Now, plants receive light and they use that energy and make, you know what they make with it? Glucose? Let's hype later. Yes, they make glucose. Or right now. That'd be awkward. So they use solar energy to fix carbon dioxide and make glucose. But look how cute this is. So look up there at the rod. The rod is the green one that's cylindrical and it looks exactly like that. But it is this long kind of cone part at the top, rod part at the top are discs stacked with pigment, just like they were in the chloroplast. So isn't that lovely? The conservation, everything's really complicated, but there's a lot of conservation of what works. So your light reception, you're not making glucose with it, you're sending it to the visual cortex, and you can see. But the process is really similar, and I just think that's divine. In fact, it is divine. It's divine, the way that works. Okay, so light gets converted to electrical signal, and the light is received by the pigment, and so you have cones. Now remember, that's the rear of the orb. It's back that way. The light is going all the way through, and that's where the photoreceptor cells are really just buried into that epithelial pigment at the back layer of the retina. And see, then there is kind of one of those, then they're scanning electron microscope things. And you can see all those layers. It's very linear, it's very organized, which makes sense because you have this pixelation of information that you're trying to coalesce and create something visual with. And also moving, we're not just looking at stills, things are moving too. So this is really incredible. So that's the way that looks. Now, rods. The pigment is rhodopsin. You've heard this before. Rhodopsin is a two-component molecule made of retinol, which is a vitamin A aldehyde. Vitamin A basically gets oxidized and is converted to retinal. The protein to which retinal will be attached to is called an opsin. And there are a few flavors of opsins. The opsin in the rod is called scleropsin. It's just the flavor. And when you attach retinol to an opsin, it denotes to the retinol what wavelength of light will excite it. So it's a function of that protein which wavelength that will be excited by, which is exactly what I just said, and I couldn't make it sound any better. So the retinol is just a protein that will undergo a conformational change. It's the opsin that kind of is the absorptive, defining wavelength portion of that. It is, if this is exciting to you, it is a G protein coupled receptor, opsin is, and it's in the cell membrane where G protein coupled receptors often are. But what's going to happen is you're going to really change what the ionic flow when you excite that rhodopsin. And so the opsin is right there in the cell membrane where you can get that started. So at rest, retinal is in the 11 cis formation, which means it has a little bend in it. That's all. And then it's sitting there stuck to its opsin, and then it will get excited at a particular wavelength. And I forgot which wavelength it is for rods, but you know, it's kind of in the green wavelength pigment, actually, green area. So there are the cyclic GMP gated channels that allow sodium in. And so that's a very used over and over kind of coupled system there, where cyclic GMP is present in the cell, the channels are open, and sodium flows in. So that the intracellular content of sodium is high. And what that does is it creates this braided potential or ionic polarity that causes that cell to release its neurotransmitter. So at rest is really a state of depolarization. What? I hate that. That's all backwards. That's how it works. And so we call it kind of the dark current. That's exciting, right? The dark current. So it's dark when it's excited, in a sense, or when it's at rest. And so when glutamate, that neurotransmitter released from the rod photoreceptor cell, does at its synapse is inhibit that cell. So we can turn that bipolar cell on by closing the sodium gates. So this is at rest. 11-cis retinol is attached to opsin. Cyclic GMP is abundant in the interior of that cell. Sodium flows in. So let's excite that cell, shall we? Oh, and see see this first little picture is the opsin in the cell membrane, and retinal is supposed to be in the center there. And then in that next picture you can see 11-cis retinal is the one with the king trans is straight, which sounds a little opposite, I think. But, and see how there are those discs, so there's lots of cell membrane for opsin to be in. So you're not just talking about just the top of that rod where the photoreceptors are. There's all this cell membrane, and so there's lots of photoreceptor cells. So this you begin to see how you create all this variation and intensity of the light, because you can have the stimulus be graded to some extent, how much retinal, how much photoreception is recruited here in this one cell, and plus the other ones around as well. So there's a lot of it in there. It takes a good bit of light to get that cell to become activated. So, this has a lot on it, because it's the whole thing. So let's just baby step through here. Light enters the orb. I'm just going to quit saying orb. No, I'm not. 11-cis retinal, gates open, sodium in, neurotransmitter out. Now light's going to hit it. 11-cis retinal straightens to 11-trans retinal, and it dissociates from its opsin. Now it's separated, so we call that, have you ever heard that bleaching? When you heard this before, people call that bleaching of the rods. I mean, I don't really care. But it makes sense in the sense that light bleaches them, because that opsin, that retinal has to be reconfigured back to its original 11-cis conformation, and that actually takes minutes for that to happen. So most of this stuff, I mean, it's just infinitesimally fast, or super fast. And that is the actual minutes. And so light travels at 186,000 miles per second, but it takes minutes for that 11 trans to get back to 11-cis. And that's also an ATPase-dependent step. So that's why it's kind of called bleaching. It takes a while for things to get back together. But we have lots of rods. Rods are really covering the whole retina except for that very center. So what happens when opsin is now without its retinal? It activates transducin. And that's the same exact thing that's written over there in that form. Transducin will hydrolyze GTP. Is this one of those things where you just go, or do you just love biochemistry? You do? You love biochemistry? Oh, thank you. And baby Jesus. And then that activates phosphodiesterase. So really, this is just one of these same old cyclic GMP, GTPase kind of little cyclic things. But happens is transducin activates phosphodiesterase. Phosphodiesterase is going to cleave cyclic GMP. Cyclic GMP now is reduced in the cell. It's because it's destroyed by phosphodiesterase. Remember those sodium gates? They were cyclic GMP gates. They were open because cyclic GMP was abundant. But now this opsin, transducin, phosphodiesterase cascade has removed that cyclic GMP. Those gates close. Sodium levels decrease in intracellular fluid. And the glutamate is no longer, or the neurotransmitter is no longer released. So that cell is shut off. It can no longer inhibit the bipolar cell. So that bipolar cell will depolarize. It will signal the ganglion cell. The ganglion cell will depolarize such that an action potential is sent along its axon via the optic nerve to the visual cortex. Believe me, don't you? Let's see. There must be another word or something I can throw in here that'll just bam. Yeah, no, not really. So what happens now that the cell has, it has to get back to its at-rest state. So the 11 trans-retinal gets converted back to 11 cis-retinal. It will diffuse back to the opsin and become attached. And cyclic AMP will no longer be destroyed, so it can begin to collect within the cell. Those sodium gates will open, and you're right back where you can then become excited again. But it takes minutes for all that to happen. For the ride. Oh, here, this picture should just put it right into perspective. So I chose a variety of pictures here because something makes sense to somebody, or something makes sense to nobody. But here's just an example. You can see starting with one light stimulant. So you see rhodopsin there. It's a, what the hell is that? Like a light channel? A light is striking it. This is a light channel. Number 2, where it activates G protein, stimulates cyclic GMP phosphodiesterase. So you're getting rid of cyclic GMP. So see down here by the sodium gate, it's going away. And then the sodium gate's closed. Yeah, that picture didn't really change it a lot, does it? But when you're sitting and enjoying this later, and you will, more than once, you can enjoy that. Choose your picture. Some people like pictures. I had to draw pictures. I had to draw cycles with my hands, or I couldn't learn things. So, you know, there you go. Oh, and here's another one. In case you like black and white. In the dark, cis-retinal. And the channels are open. There's no action potential or gradient sent because, oh, that's backwards. Oh, that was the bipolar cell. Whoops, skipping cells. Okay, so you see neurotransmitter is released, bipolar cell is inhibited, ganglion cell is inhibited. In the light, excite the bipolar cell, excite the ganglion cell. Light sends the electrical stimulus to the visual cortex. There you go. Cones. Photoopsins, this is another word for opsin, but there are 3 flavors of opsins for cones, and they recognize light in various wavelengths. The activation pathway is really the same. It's just what wavelength of light excites that cell. And so for rods, they need low light, but there's no color. So it's all grayscale. But low light. Cones need a lot of light, but they can also see color. And so that's the only real difference. The activation is the same. So there are three kinds of cones: L cones, M cones, S cones. And L cones receive long wavelength light, or they're the red cones. Red light is long wavelength, it's kind of slower. Short wavelength is faster, blue cones. And you see that there's a lot of red, a little bit of green, and or so half as much green and 2% S cones. So here's the wavelength of visible light. We see in that 400 to 900 nanometer wavelength of light range, because we have cones and rods that can see in that range. And the rods are kind of in that it's like 496 or something, or just over 500. It's around 500, and so their light would be perceived as green, except we perceive it as grayscale. Their response is faster, and they can kind of get back to their original or resting polarity a little bit more quickly. So, how do you then perceive? It's this red, it's the primary color thing, and that you can take primary colors and blend to make other colors. And for the life of me, I never remember them because I don't know. Red and blue make something, and then yellow and I don't know. But you know. But anyway, that's how it works. I'm not going to ask you how do you get orange. I mean, what percentage of this count and that count. But that's kind of how it works. And so that what is it? Come on, one of you know. What makes red and
Purple.
Okay, this still doesn't matter because I can't remember. I don't know. I have a block against those things. But anyway, this is how you see color, is that it's a blend of more than one cone activity at a time, and how much of red and how much of the other one, whatever, and so you get this blend of colors because you only have these three things to work with, but there are a lot of them. And so the intensity, what amount of recruitment of green and red, if you looked at something just red, then you see it very intensely as in red and just those cones, but other colors begin to blend in there, and you work your cones until that color comes off with something. And so when you think about it, it's all kind of arbitrary. We're just looking at bounced back light, and we perceive it as a certain color. So what does that make the world really? I don't know. That was the existential, if you know what that, as you did. Color theory. Did you ever take any of that stuff? You learn color theory, and they can just ruin color for you. That's why I don't remember these things. But there are really three things about color. And this is really how three flavors of cones work. But when you think about it, it does make some sense because there are only 4 DNA nucleotides, and yet we make all the diversity in life, right? So 3 colors of cones make all the diversity of color. So hue, saturation, and intensity. And I really kind of liked this little wedgie thing. Oh, I don't like wedgies. I like giving them now. Okay. So hue is the actual color. Saturation then is brightness of color. Is that what I want to say? And then saturation is intensity of that color, and value is brightness of that color. And so if you ever messed around, with your photo editing, now they're already photo edited. You just pick your one or whatever. But if you go to any of those, and you get to choose contrast and hue and saturation, you see the difference. You can increase hue and make yellow look orange, it's interesting. So anyway, that's how those 3 cones can create all of this in this room that you're never going to get out of, and this is all you're ever going to look at for a year, isn't it? I'm meant to be more positive. And then you see the wavelength, blue, short, red, long, and then there's the cross-section in between. So anyway, and then remember there were rods. There are 3 flavors of cones, and then there are the photosensitive ganglion cells, and they're just detecting light, dark, and here's how that works. It's similar, a melanopsin kind of thing, a melanin plus an opsin, and it's just detecting light itself, and you're not perceiving it in the visual cortex, so it signals the hypothalamus really for circadian cycle. And then also, the parasympathetic nervous system can change pupillary reactions, so that pupillary reflex. You know, that's not voluntary, that's reflexive. And so your pupils dilate or constrict. So at rest, constriction is more where parasympathetic is going to fit them. And then if you get excited or emotional, you want to let all the light in so you can see all you can see when you get this big pupil. And so that really, that information is kind of coming from these photoreceptive ganglion cells. Cute, right? And then I have a slide next that has a lot of words on it. Did those, oh, people put those damn slides up that had like a graph and a figure and all those little things, and they freak out, and then they're going, that's what you need to know. I mean, I'm sure you'll get none of those. The cardiologist would do that a lot. Okay, there's a lot of words here, but mainly I just want to put that circadian clock thing in here because it's not a function of vision, but it is really a function of visual neurons. And so the clock in mammals is in the suprachiasmatic nucleus in the hypothalamus. So that's the same thing as being in the hypothalamus. The signal from the ganglion cells is sent along a retinohypothalamic tract, which is cute. It goes from the retina to the hypothalamus, so it's called the retinohypothalamic tract, like it. And it interprets and passes it on to the pineal gland, which is located sort of on the top of the thalamus. And then the function of that is to inform the body by secreting melatonin. And so, you know, that's the thing that you can take to help you kind of sleep. Melatonin is secreted at night and it ebbs during the day. And so that helps set up our light-dark cycle for this 24-ish hour rhythm we have. So it comes from information received visually through, not visually, but through the eyes. Yes? And turkey. Oh, God. I'm just going to say this. Remember there are 150 photoreceptors, but only 1 million ganglion cells. So there's a lot of information at the retinal layer that gets kind of squoze down to something that gets sent to the visual cortex via the temporal. And so how that's kind of done, what this is sort of figured out to be like, is there's a lot of packed concentric kind of information through these circles. So there's an interior circle and there's an exterior circle, and sometimes there's another little layer there. These kind of like little donuts of rods stacked together or cones stacked together. And so, and really, how would you arrange that if you were making a retina? Really? I mean, how would you arrange all of that so that red gets sent and then you can make purple or however you do that? Or, you know, would you arrange that? And so there are these kind of like little discrete packets in a sense that can see information. There's an interior and an exterior. And so center cells send their information directly to the ganglion, and all this surrounding information is going to be inhibiting it or exciting it. Basically, it's something like that. So there are these on-center and off-center, and you can just Google to your little heart's content, but it does not increase my understanding of how that works because it's so incredibly complicated. 150 million receptors in a retina. So you've got as many on that other side. And you have a mosaic, basically, of pixelated bits of information that is coming in these little centers, on, off. So you see how they're showing you some cones there. Several cones are feeding or converging onto fewer bipolar cells and even pure ganglion cells. And this stimulus and where something lands in that circle is how that works. I could find no reason to continue to go into that or show you more pictures of that. It's just miraculous. How do you send that information, that mosaic, that complicated mosaic of information in? How do you integrate that and then send it and visualize anything? You go from them, their eyeballs, they're bilateral, right and left. The optic nerves are bilateral, and they meet in the middle there at the optic chiasma. You know, chiasma means cross. And so some fibers from the right and some fibers for the left will truly cross there, so that each bilateral visual cortex is receiving some information from its contralateral eye as well. Or we would have two monocular visions. We have binocular vision. We see different things with each eye, but there is an overlap, and that allows for depth and for dimension. So we can see things in a 3D sense, and we can see where they are in space away from us as well, because of this convergence of material between the two sides. That information follows the optic tracts back to its own visual cortex via the lateral geniculate nucleus of the thalamus. So you studied, oh yeah, sure. Oh, you just wanted to move, didn't you? I do too. I'm just going to move around this time. Lateral geniculate nucleus of the thalamus. You know, the thalamus is it's paired, but sits centrally located, and really is a relay station. Now, you did all this structure, but you really haven't done your neurology yet, so there's not a lot of function, right? So whatever sits in the middle is really going to relay stuff to the two sides. And so your brain stem comes up, and then you have the thalamus there and hypothalamus, and then the corpus callosum will lateralize the two hemispheres. So the thalamus is bilateral, but it's really serving as a relay to get stuff to go where it's going. And so you're sending stuff back to this occipital cortex. The higher the information goes, then the more consciousness and awareness we have of it. So anything that's just going as far as the brainstem, I'm pointing at my brainstem. Did you recognize that? Back here, this way. That's very reflexive. You just don't have much conscious awareness of it, right? The higher you get up, the higher it goes. The thalamus is really important. Everything goes to the top. So much that goes to the top, to the cerebrum, is relayed through the thalamus. Amazing bundle of tracks going through here. And so, because vision is going all the way to the cortex, we have this super conscious awareness of what we see. We relate it to memory for what we've seen before. We can remember the actual picture. We can remember the actual color, which is one of the hard things to remember. And have an emotional reaction to it. The more your emotional reaction is to it, the greater your memory is of it. You know all of that. And yet, you don't need to be able to see. It's not essential to function. I mean, it helps you navigate the world in a survivability way, but you don't need it. So that we've dedicated a lot of brain to that, so that's why that's kind of a higher thing. And yet later you're going to be playing some stupid game just for hours, and there's so much else to look at. So look, visual cortex, quite a bit of cerebral hemisphere is dedicated to this. Just, it's miraculous. Okay, so here's just another kind of way of seeing where it's all situated through that guide. From the eye to the thalamus, right and left, showing you where the hypothalamus is there. So light falls in the retina over here, you can see it going through the eyeball orb, back along the optic tract, kind of up to the chiasm, and then out to those lateral, kind of up and out to the lateral geniculate nuclei, and then on back to the visual cortex. Now, then what happens, no one really knows.
Do they cross? So
Here you go. Perfect timing. There's this cross of information. So look at this. This is interesting. bilateral eyeballs. There is a temporal bit of information that comes from that temporal retina, and then there's the nasal side of the retina. So there's the retina is kind of divided into these two halves, the visual field that they see in here. Remember, if everything is also turned reversed and upside down, and so there's just all of this business that has to happen. So we might actually all be upside down right now, I'm not really sure, it's freaky. So you have this information coming from these two halves. So the temporal half stays with its own side. So the temporal half of the left eye is received to the visual cortex of the left hemisphere. The temporal portion of the right eye is received to the visual cortex of the right hemisphere. But there is decussation or cross between nasal visual fields, or I should say, I can't make arms do that. Nasal visual fields. So you see that in the right eye, the right nasal field will cross at the chiasm and go to its contralateral thing. It's interesting because what you're able to do there then is have lateralization of vision. You don't have two monoculars, you have binocular vision. And that just makes sense, that you would send fibers from each eye to the other side and to the same side so that you can lateralize that information. Because this isn't really going through the corpus callosum. The corpus callosum is sending information to the hemispheres. You're going straight from the source, the receptor, to that visual hemisphere, to that visual cortex. And so you do have that. And what happens is some really bizarre and cool and fascinating deficits that I don't want to go into here because you're going to talk about them, I think, with tomorrow in your pathophysiology kind of stuff. But what really happens, I worked with strokes for a long time, and you have a stroke or ischemia on one hemisphere, and so you have motor loss on the contralateral side. However, you get some funky chunks cut out of vision because you have this, people would lose both like nasal on one side and temporal vision on another side. And there's just a lot of things that physical therapists really are the ones spending time teaching people how to deal with this deficit. But I don't know, maybe you've seen these things where they turn the plate around so they can see the other half. It's not like they can't see it, but they don't know where, they can't interpret it because maybe that part of vision is lost or visual interpretation is lost. So if you move that field to them, you come up to them and they have no idea, hi, and they're just looking that one way. And then you go, hi, and they'll go, hi. Because they neglect that side or can't see that chunk. So those are some hemianopsias or some visual field losses, but it's not just right and left. It's right and left, right and left. So it's really neat, and you actually can assess that in patients with when you confront them, when you see my fingers, or which eye did you see it, or they'll have to turn. Strokes are a fascinating way to learn about the nervous system because you see the deficit. And when you're choosing your rotations later, I mean, I have no reason to sell you on a rehab rotation, but also if you go to, geriatric kind of settings, you'll see the deficits in stroke are endlessly fascinating, and only then can you understand all of the stuff you're learning, So this is really the part. Keep hanging in there and keep coming back, it gets better, because you see the deficit and then you'll forever understand it, Anyway, but because there is this crossing so that you can bilateralize information, you can have some really interesting loss when it comes in the brain, and the losses in the brain. Okay, so when you get back there, when you get back there to the visual cortex, so there are the eyes and the nasal and temporal fields, and then the optic chiasm. Let's see what I want to say. The lateral geniculate nucleus that in the thalamus. It has all these layers and parts, and this is how it's able to send and help interpret. So really I'm just saying relay occurs of this information. Because at this point, the information is only an electrical stimulus. And an electrical stimulus is only polarity. Either it's positive or negative. This is endlessly weird because somehow when it gets to the cortex, you have a whole vision and memory and reaction, and that crap is moving too. So it's no one gets it. But there are layers, and we can have areas of function and try to begin to understand it. So then there is this, to actually get to the visual cortex from a lateral geniculate body, there is this geniculocalcarean tract. So I'm just kind of showing you further how you go from thalamus to visual cortex. But look at this. Okay, don't freak out. Okay, so don't freak out. My husband, we always have a big dog. And he goes, calm down, boy! I just think that's so funny. Every time he yells at him to calm down, I just think, serenity! Makes no sense, dude. I just like, it's not working. He's going, oh, I'll give a ball. Okay, look at all these layers in the visual cortex. V1 and V2 and V and 4AB or whatever that says. Anyway, it just means somehow we are sending these million ganglion cell axons, then get to the thalamus, and then they get back here. And you're relaying bits and portions to all of these different things so that you can interpret it. So there's all the pretty layers. There's lots of layers or areas of function in the visual cortex. And don't freak out, but this, I don't want you to know this at all, but I just wanted to say, because I think it'd be silly just to go, then the visual cortex can realize it as sight. As you go, there are chunks for understanding different things, and we're able to collate that information as vision. So you see, primary visual cortex is V1. Let's just look back where that was, just for curiosity. It's green, it's big, it looks like more space is dedicated to it, because you know this cartoon is true. Also, I got it on the internet. It begins processing, and then there's relay of information. It's not like it just goes and sticks to that part and that's it. There's all of that interconnection in the cortex and all this, the whole rest of the cerebrum too. So they're sending information around so that you can understand it. So large visual fields, motion, detection of shape, color, texture. Oh God, texture. I mean, really, there's a lot to this. And movement and speed of bodily movement. Somehow they've sort of said, well, if you have this missing, you lose this part of vision. So that's how some of this functions are identified. Upper and lower quadrants, color vision. So what am I telling you here is I don't know how any of that works. And that's what I'm saying here. My very favorite, what's your favorite thing to look at? I mean, don't tell me. I don't know. You look like, I'm thinking porn. For my porn is Norway, pictures of Norway and the Aurora Borealis. I can look endlessly at pictures of this because I will never see it because I don't like to be cold, and it's really the best time of the year to see it is cold, and you're above the Arctic Circle. So anyway, can you imagine seeing that? So anyway, what I'm just saying is visual cortex, all that stuff is processed, and then you can see it. But you don't know how, but it's pretty. It's pretty. Sometimes it moves. That's it. And then the next section, woohoo! This is a really long lecture. They don't have me scheduled a lecture in a 4-hour chunk ever again. So, go pee, and then we'll start in 5 minutes, and then we'll do hearing. Do you have any questions you can think about, other than, and asking me to stop is not going to? You had a good time? Water yourself.